Good evening, everybody. It's a great pleasure to welcome you to Professor Declan O'Regan's inaugural lecture entitled Machine Intelligence in Cardiac Imaging. Declan O'Regan is Professor of Imaging Sciences at Imperial College. He's based at the MRC's London Institute of Medical Sciences and has run a group there for many years. Declan's research really is at the nexus between genomics, imaging, cardiovascular science, and AI. He's won a great deal of awards from numerous bodies. He was the Roentgen Professor, Travelling Professor a few years ago, and he sits on the Advisory Council for the British Heart Foundation. He's a superb colleague and a great friend. Before I pass over to Declan, I just want to remind you of a couple of housekeeping elements. So you can submit questions about Declan's presentation at any time in the Q&A box. As the event is being recorded, can we ask that if you don't want your name read out, you submit these anonymously? And if you like a question that one of your colleagues has asked, maybe you'd like to give it a thumbs up. So without further ado, great pleasure to welcome Professor Declan O'Regan.
Mandy, that's great. Thank you very much indeed for the kind introduction and for all your support at the LMS. So it's a great pleasure to give this talk on machine intelligence in cardiac imaging. So this hierarchy probably be familiar to many people about how AI is defined. So the conventionally thought of a machine's ability to generalize learning in order to efficiently achieve quite complex tasks autonomously. And we might think of things like driverless cars, for instance, in that category. Machine learning is a broad category of algorithms to improve task performance without needing to be explicitly programmed. And that might include paired input and output data, or just unstructured data where we want to find patterns in it. And then down to deep learning, which is a form of multi-layered neural networks to progressively obtain more complex and more useful representations of data. But actually, I would argue that it's not the code that's important, really. It's really down to how data can be best used to prevent and treat disease. And the sorts of algorithms, the sort of architectures I've touched on will change over time, but the fundamentals won't. And the fundamental drivers are really how we can better treat our patients and better recognise disease at an earlier stage. So I want to slightly reframe this a little bit into more of a task-orientated view of what we're trying to achieve. What we're trying to do is model the true health status of patients, not just from outward clinical observations, but by integrating all the available health data that we have from very diverse sources now. And that can include imaging and biophysical models, but also data from electronic health records and so on. So if you just rely on observing typical symptoms of disease, it may well be too late to intervene effectively. So for example, having a genetic risk factor could be an opportunity for appropriate risk modification. And then as the heart moves from to a compensated state, and that might be the interaction between a genetic variant and environmental risk, there's a chance for preemptive detection and surveillance. Then in early disease, there is a window for intervention before disease progresses and may be irreversible. So I think this sort of view really where we have increasingly rich data on which to model the state of the heart is really valuable. And this is where I think imaging can really come to the fore, because particularly cardiac MR, because it makes the heart visible to us. So it allows us to integrate all of these risk factors, these clinical signs and symptoms and biomarkers, but actually it makes the heart transparent to us. The challenge really is how to quantify all of those features and to make predictions about the future health state. So I'm going to reframe the sort of first figures that I showed you about that hierarchy of AI and put this in the context of machine intelligence in heart disease. So we're not trying to imitate what humans can do, but instead we're trying to perform tasks that humans can't do well and try to add value to healthcare. And that involves integrating imaging with genetic data, with other risk factors and biomarkers, and trying to build a digital simulation of the heart, really across diverse populations, and importantly, trying to make actionable predictions in order to guide treatment. So much of what I'm going to talk about revolves around MRI, because MRI is such a wonderful technique for looking at the heart. So this is a one-slide summary of how the principles of MRI. Of course, it involves a magnet, a large superconducting magnet, and you put the patient inside this magnet. And what we rely on are the protons. We're interested in protons, and in a strong magnetic field, the protons, particularly abundant in fat and water, they separate into two energy levels. The stronger the magnetic field, the bigger the difference there is between those two energy levels. Now we can push the protons from that low energy state into a higher energy state by adding some energy at just the right resonant frequency so that it can be absorbed. And we can do that inside the magnet using an oscillating magnetic field. Then those protons fall back down into the lower energy state and they emit a radio wave. And we can collect that radio wave just by putting a receiver coil over the chest. And we can use additional magnetic gradients in addition to the main magnetic field to slightly change the strength of the field in different directions so that we can localize the signal and build up an image of the heart or any other structure in 3D. Also, of course, different tissues have different magnetic properties and relax back to that ground state at a different rate. And that gives us the contrast between different tissues or between normal and abnormal tissues. So the history of MRI is quite long and complicated, even a little controversial. But these were two of the key players in MRI, Paul Lauterbur and Peter Mansfield from Nottingham. They didn't exactly have linear approaches to research. Paul Lauterbur worked for the inventor of silly putty, and actually was the first imaging was done to characterize rubber substances. And Peter Mansfield, before he went to Nottingham, he left school at 15 and started work as a printer's assistant. It didn't stop them both getting Nobel Prizes. And the first paper on MRI was published in 1973, and that was on a phantom, but it was only 4 years later the first image of a human body part, it was a finger, was published, and that was by Sir Peter Mansfield's group in the BJR. Now, we do say that, you know, sometimes your equipment belongs in a museum. So the image on the left is, in fact, the scanner that was here at Hammersmith Hospital and is now in the Science Museum. So this was, in fact, the first whole body MR machine of its kind in the UK. And now is a picture on the right there of the current scanner that we have. But the principles are essentially the same. And cardiac MR is really hugely versatile. We can examine the shape and motion of the heart. We can look at tissue characteristics, flow. I've got some examples of three-dimensional flow that you can see in the bottom right there. It avoids radiation. It's very safe. It's very reproducible and very, very comprehensive, the assessment that we can make of the heart. And it's the perfect substrate from which to build a population model of the heart. And UK Biobank has just been transformative in this. So they have the aim of recruiting 100,000 people for MR imaging. They all have health outcomes and genetic data. They've got to about half that target already. And it's really a transformative resource for research. And much of the work that I'm going to be touching on is going to involve UK Biobank. Okay, so how did I get into radiology? Well, I suppose it was a bit non-linear myself, really. Probably most people don't know that I actually wanted to work in tropical medicine. So I had a particular interest in that. And after my house jobs, I went up to Liverpool and studied for the diploma in tropical medicine and hygiene. Liverpool was amazing. It's a fantastic place, particularly on a Saturday night. And of course, it was where all of the merchant navy and seamen returned from the tropics with fevers and so on. So that's why there's a Liverpool School of Tropical Medicine. And I really wanted to develop a broader understanding of health beyond the UK as well. It was very interesting for learning a bit of epidemiology, for pandemics. It'd be interesting to go back and see if anyone mentioned coronavirus. I bet they didn't. They also had a vivarium in the loft full of snakes that they used to milk every day to get the venom so they could get the antidote. And the hygiene bit, there was a lot of teaching about how to dig latrines, including a field trip to Wales to learn exactly how it's done. In fact, I think that the mantra was, if you're in a refugee situation, it's much more important to put down your stethoscope and pick up a shovel. So I haven't forgotten that message. So from then I went on to do medical rotations down in St. Richard's in Chichester. Fantastic place. I think it's the only doctor's mess I know of that has its own yacht. So it's a brilliant place, brilliant colleagues. The flu season there, 1999-2000, was one of the worst in 20 years. We have quite an elderly population on the south coast. It was quite an interesting experience. I suppose no one could ever imagine what was to come with COVID. But although it was a fantastic experience, I did decide at the end of that acute medicine wasn't going to be for me. So I decided to apply for radiology, knowing very little about radiology beforehand. Came to Hammersmith Hospital in 2000 to start training. And then actually by chance, volunteered for a scan in what was then the Robert Steiner unit. And that's where actually the magnet that I showed you from had been installed back in the 1980s. And it was really a huge hub for some of the very earliest steps in MR that have been made since the 80s. And it still had a very thriving research program, particularly in MR physics at the time. So I was persuaded that it might be a fun thing to do a PhD. So we decided to do it on fat, and actually this is a histological section of the liver, and the white blobs are areas of fat deposition, and you can see some scarring in the liver as well. So the question that we were interested in, and it's still a really topical question, is understanding mechanisms of fat, how fat builds up and how it damages organs, and particularly what's the relationship between fat in the liver and fat in the heart. So non-alcoholic fatty liver disease, or NAFLD, affects maybe even up to a quarter of people now, and it is reversible, but it can progress onto an irreversible state of hepatitis. And it's characterised by inflammation and fibrosis, which can lead to liver failure. Now, this is all part of deranged metabolism, a pro-inflammatory, pro-fibrotic state that is partly mediated by the liver and also by visceral fat, so fat that surrounds our organs. What wasn't really understood was whether the heart was part of this story and whether fat has a toxic effect on the heart muscle, what effect it has on diastolic function, that's how stiff the muscle is, and also on the coronary arteries. So how are we going to see how much fat there is in the heart? So I mentioned that when those protons go into the scanner, they divide into two energy levels. Those two energy levels are slightly different for fat and water due to the molecular composition. So it means that water has a slightly higher frequency than fat. So like these two tuning forks, if you have two tuning forks that are slightly out of pitch with each other, they interfere. So there will come a point where the frequencies add up constructively and also destructively cancel each other out. And so you can use that same principle for trying to quantify the amount of fat, because if there's equal amounts of fat and water, then the two will cancel each other out. So Stefan Schmitz, who I think is here this evening, he was my PhD supervisor, super enthusiastic. He was a fantastic person to really give me an introduction to research. So essentially we use this technique to build up maps of water, of fat, and of T2 star, which is a sort of measure of iron content. And you can see in the graph at the bottom, you can see how this is wavy. So this basically reflects that sort of acoustic beats phenomenon, the fat and water signals cancelling each other out. So if you model this, then you can figure out the ratio of fat to water, and you can also figure out how much iron there is in those tissues as well. We tried to do this on the heart. It just didn't work as well as we'd hoped. There's very little fat in the heart, and the amount of motion complicates that as well. But with Stuart's support, Stuart's been a fantastic friend and colleague and advocate for my research. We decided that we would actually look at the T2 star bit of this, so actually trying to quantify iron in the heart. And there'd been a lot of work already done looking at patients with thalassemia. Thalassaemic patients receive multiple blood transfusions and they get iron overload, and the iron is deposited in the liver and the heart. So there's been some really nice work looking at quantifying the amount of iron that's present in the heart muscle using this T2 star sequence, which is very sensitive to the presence of iron. But we had noticed on imaging that patients who'd had a recent infarction and had been treated for that, they seem to show some signs of iron deposition in the heart as well. So when someone has a heart attack, when the coronary artery is blocked, all of the heart muscle beyond that becomes ischemic, so it's starved of oxygen and nutrients quite quickly. Then it becomes edematous, so it starts to swell, but it doesn't die instantly. So there's this wave front of necrosis that sort of progresses outwards. And the idea of treating a heart attack is to re-establish flow as quickly as possible, prevent that wave front from passing through the whole of the heart muscle. But even the act of reperfusing blood to the heart muscle, although it's very beneficial, it can also cause damage to that muscle as well, and it can cause bleeding inside the tissue. And where there's bleeding, there's iron, and that's something you can measure with these T2 star sequences. So where the arrow is shown here, the red areas where there's a particularly high concentration of iron within the muscle, and that corresponds to where the infarcted tissue is as well. So I think this is really interesting, and it's still an area of active work, understanding both the benefits and potential harms that reperfusion injury can do to the tissues, and how MR essentially can be a sort of imaging biopsy that gives you an unprecedented, unequaled access to understanding the properties of the heart muscle. So the next project that we're involved with was really thinking about the limitations of the way we currently analyse heart images. So here we have software that analyses the moving images of the heart, and it generates numbers like the maximum minimum volume of the heart, an ejection fraction, and the mass, and so on. And these are very sort of well understood, validated sort of metrics, but they're very insensitive to the complex physiology of heart disease. So could we start to think about a way of really modelling in much more a realistic way the structure and function and motion of the heart? So we developed a 3D sequence. So this was a single breath hold sequence. It captured the whole heart in about 15 seconds or so. And with Daniel Rucart's team at South Ken, we developed computer vision techniques to automatically analyse these images, to label the heart muscle and to track it over time. And we actually did a mini biobank study. We recruited nearly 2,000 healthy volunteers over a 5-year period, and genotyped and sequenced them all as well. So it's a fantastic resource that we still use for validation studies now. But the principle is that you can analyse this data and then you can represent it in a standard coordinate space, in an atlas, so that every patient is in the same sort of spatial coordinates and is very, very consistent. So then you can apply shape analysis and statistical tests on this single 3D representation that captures the variation in structure across a whole population. And that allows us to tease apart the relationship between, if you have a genetic variant, how does the development of the heart change? How does it respond to environmental stress? What are the very earliest signs of disease that might not be evident using conventional imaging? So Antonio, who will hear from later, is a fantastic friend and colleague who's been working on these data for many years. It's back in 2015. One of the things we wanted to look at was the precursors of hypertensive heart disease. We know in hypertension, the muscle of the heart becomes thick, and that is an independent predictor of poor outcomes. But this data here actually showed that those initial variations, those initial responses to hypertension actually occur way before people are diagnosed with hypertension. So even in a very low risk population, there's still this relationship between increasing wall thickness and blood pressure, the same sort of pattern as you see in people with hypertension. And this is really some really nice work showing how the heart can have maladaptive responses to high blood pressure at a very early stage of disease. But actually taking that same concept forward, some very recent work that we've had done you can actually translate this into predicting the risk of carrying a genetic variant, what that might be on your heart. So hypertrophic cardiomyopathy, it's an important cause of sudden cardiac death, and it's associated with variants in the sarcomere, variants that or genes that control for the development of the sarcomere. The sarcomere is like the basic contractile unit of muscle fibers in the heart and elsewhere. And it's associated with abnormal thickening of the heart muscle, but also developmental abnormalities in the heart and fibrosis. All of these can contribute to an increased risk of sudden death and arrhythmia. But it's normally a disease that's seen in the context of affected families. So one thing that we thought was important to look at was, what about the risk in the general population? So if you or I were to have a genetic test and it was to come up positive for one of the variants we know that we see in families with HCM, what would that mean for our own risk? So, and what does your heart look like outside of people with established disease? And this was a great collaboration with James Ware and Catherine McGurk and Antonio again. So we found a pretty conservative estimate was about 1 in 400 people carry variants thought to be pretty definitive or likely to be disease causing for HCM. Much more commonly, almost 1 in 40 people carry a variant that we're not sure about yet, that may or may not cause HCM. And if we look at the age-related association with adverse events, we can see there's a strong increase in events as people age, but in those that carry these pathogenic variants, these variants associated with HCM, there's a significantly increased risk of death and major adverse events, about a 69% increase. For heart failure risk, it's about 4 times that. Importantly, though, we found that mostly the majority of people lead healthy lives. So they have no, the absolute event rate is quite low, but there is an increased risk of developing these complications. So what does the heart look like? So the interesting thing is that these same genetic variants that we know cause overt disease in HCM families, those same genes seem to cause a very attenuated form of HCM in the general population. So you can see the 3D models on the right, the areas in red are where the heart muscle is thickened, but in most cases, 80% of cases, it wouldn't be enough for you to be given a diagnosis of HCM if you were to be seen in the clinic, for instance. So why is that? It's probably because there are common variants that are also inherited in families that affect the expression of these rare genetic variants with larger effects, and there's a not co-inherited in the general population. But I think this is particularly important now with the growing availability of genetic sequencing, and also throwing up these incidental findings when people are screened for another reason. It's important to be able to counsel people about the potential risk and to put it in perspective as well. We've done some similar work on dilated cardiomyopathy. We've got an example here, a post-mortem example here of a patient's heart who's died from dilated cardiomyopathy. You can see the chambers, the heart are enlarged, they're very trabeculated, with muscle fibers very evident, and it's a leading cause of heart transplantation. And here we've got the sarcomere. This is the contractile unit of the heart, and titin is this protein in green. It's sort of like a molecular spring, and it's important for sensing tension in the heart, but also for its elastic properties. And important variants in the gene that encodes for titin are found relatively commonly in dilated cardiomyopathy. But again, how does this disease arise? What if you or I were to carry this variant? What effect would we expect that to have on the heart? So this is work that we did again in our population of 2,000 healthy volunteers, carefully screened to have no history of cardiomyopathy or no family history of cardiomyopathy either. So what the models here show, we've got one in diastole where the heart is relaxed, one in systole where the heart is contracted. The red colours show the effect of carrying that variant on the shape of the heart. So the stronger the colours, the stronger the difference in geometry of the heart. So there are definite structural changes if you carry one of these variants, even though superficially these patients' hearts look relatively normal. And taking this with other data from animal models, it suggests that if you carry these variants, basically your heart is in a sort of compensated state, but it's inflexible to any further stress that might be put on the heart. And that can include a variety of things, like alcohol or chemotherapy or pregnancy or myocarditis and so on. So it's been a very useful way of understanding what the precursors are to disease and the interaction of carrying a genetic variant with an environmental risk factor in pushing you towards a decompensated state. What about predicting outcomes in heart failure? This is a key thing, and you know, I think as imagers and radiologists particularly, we focus a lot on diagnosis. What we're probably relatively poor on is prognosis, but it's risk stratifying patients and predicting future events that really drives management decisions. So here we have an example of patients with pulmonary hypertension, which is a relatively rare disease, but it causes right-sided heart failure, increased pressure on the right side of the heart, and the heart eventually will decompensate. So we measure the function, the pump function of the heart with ejection fraction. In this case, these two patients both had the same ejection fraction of 22%, yet they had very contrasting outcomes. But looking at the images, there are differences visible to the naked eye in the structure, the function, and so on of the heart, but they're difficult things to quantify. So our aim here was to train a machine to learn what features are going to be most important for predicting outcomes. And this is some work that my colleague was a PhD student and a lecturer, Tim Dawes, and Galeb Bellows, a brilliant statistician who worked on this, and Yiming Duan, who was our computer vision specialist. So it was a real fusion of a number of different specialties to pull this idea together. And it hinges, of course, on computer vision enable, so that you can track every pixel in that grayscale image, convert it to a label that means something anatomically, and then create a 3D model of the heart. So this is the 3D model. So remember, we've scanned all of these patients individually, but we want to represent this data in like an atlas coordinate space, so a standard space. So the model actually shows the surface of the right ventricle, and at hundreds of points, we've got the motion of the heart as it moves inwards and as it relaxes outwards. But this model represents the variation in function across a whole population in a single space, and that makes it ideal for training algorithms because the data is very consistent. So here, for example, we've used an algorithm called 4D survival. This is essentially it reduces the complexity of data. So it's shaped like a bottleneck. So you have a lot of data coming in, but we're trying to find a smaller number of features that are going to be useful for the task that we're interested in, and that's for predicting outcomes. So if we look at the survival plots at the top here, we've got the stratification between high and low risk patients, so the probability of survival over time. For the conventional MR parameters, there's a much greater discrimination between the high and low risk people when we use the 4D survival algorithm for doing that. And that's because it's picking up lots of rich features that are probably difficult to appreciate with the human eye and certainly difficult to measure, but contain a lot of valuable prognostic information. And it's important to remember this is completely automated from start to finish. So the patient has their scan. Even before the scan is finished, those images can be analysed. The algorithm can make a very specific prediction about future outcome for that patient, which is then available at the point of reporting those images. And particularly in pulmonary hypertension, it's very much a risk-driven management strategy. So the patient's probability of dying within 12 months determines how aggressive the treatment should be. So this is now part of an NIHR and NHSX funded study with the University of Sheffield to really drive this towards use within the NHS. And we're also evaluating this in different types of heart disease and ischemic heart disease, for instance, as well. But this, I think, is something that has the potential to make a real difference to how we manage patients. What about new ways of diagnosing disease? So I've got an example here of 8 different diseases. We've seen hypertrophic cardiomyopathy, we've mentioned dilated cardiomyopathy, we've got arrhythmogenic right ventricular cardiomyopathy up here, we've got Takotsubo, which is broken heart syndrome, we've got systemic diseases that affect the heart, we've got other inherited diseases and pulmonary hypertension again. So the measurements, if you just relied on measurements to be able to differentiate these diseases, it wouldn't be very useful because there'd be a lot of overlap. So could we actually train an algorithm to do much of what a human expert does, and that's discriminate between these diseases based on the form and function of the heart. So you'll recognise this neural network. It's very similar to the one that we use to predict survival, but instead of predicting survival, we're trying to predict a disease class this time. So we input those 3D models, those moving models. We try and reconstruct those same models with as fewer features as possible, but features that are good at discriminating between different classes. So why is that important? Well, if we go back to the data from UK Biobank and we look at these 3 groups of people, the SART neg, these are the ones who don't have a genetic variant that's associated with disease. Over here we have the ones that have variants that are probably pathogenic, or probably causative for disease, or likely to be. And here we have the diagnostic thresholds for how thick your heart needs to be before you'd be labelled as having this disease. You can actually see that the ones that have that don't carry the gene, many of them would be diagnosed or overdiagnosed with having disease, and actually many who do carry the variants would be missed because the majority of them haven't developed disease. This is in the particular case of UK of community-based data. But of course, if we look at a disease case, there are lots of rich features in these images. It's not just about wall thickness. It's about the pattern of wall thickness, the distribution of it. There are other geometric changes in the heart. There are patterns of scar and so on. There are many, many features that could be very useful for classifying these diseases. This is some work that our PhD student Carlo Biffi did on an automated approach for diagnosing disease. So if you consider these 2 groups of dots, basically this is a 2D projection of complex shape features, essentially, that will that best discriminate between healthy volunteers in green and those patients with HCM. So this doesn't rely on simple measurements. What it's trying to do is to capture those global changes in shape and motion to better distinguish between these 2 classes of disease. So, and the further apart, the more dissimilar they are. Now, this is a generative network, so you can actually move through this space, and the movie on the left shows what it would be like to follow the direction of that arrow. So from a healthy person to a patient with HCM. So you can get some sense of the differences between those 2 groups. But I think there's a really interesting future in this concept in actually kind of reconsidering how we classify diseases, moving away from these slightly arbitrary and poorly discriminating thresholds towards a much more meaningful global assessment of the heart, which is really what experts are doing anyway. That could be very powerful for distinguishing different diseases that can appear superficially similar, like athlete's heart and dilated cardiomyopathy and hypertension and hypertrophic cardiomyopathy can be difficult to distinguish. And I think this is a really exciting way of improving how we diagnose patients. What about diastolic heart function? So we've focused quite a bit so far on systole, sort of like the pump function of the heart. But diastole is the complex. It's not just a passive relaxation of the heart, it's a series of molecular and electromechanical processes that initiate relaxation and promote efficient filling of the ventricles. So impairment of these very coordinated mechanisms is related to increased stiffness of the heart, and that can lead to diastolic dysfunction. And it's well recognised that reduces quality of life and leads to higher mortality. It's also, importantly, a hallmark of cardiac ageing, which is known to occur through multiple profibrotic and energetic pathways. But we know very little really about the environmental and genetic causes of that. But Biobank again allows us a real insight into this, because we have very high temporal resolution MR images. We can use these computer vision approaches to localize the heart, to track the borders of the heart and measure strain. So that's the amount of thickening that the heart muscle undergoes. And we can take the first derivative of that to basically look at the gradient of our strain curve, and we get a strain rate. So we can get a measure basically of how fast the heart relaxes during the early part of diastole, and that's a good indicator of how stiff the heart muscle is and indicates the degree of diastolic function. So we can use that as a trait, as an image-derived trait, and we can use that as in a genome-wide association study so that we can see what common genetic variants explain differences in diastolic function. So it is a heritable trait. We've found that it's causally related to heart failure. So it's like a substrate, genetically determined diastolic function is an important substrate for the development of future heart failure. And some of the genetic pathways are important to biomechanical stress and how the heart responds and adapts and repairs itself to stress. Also a potential target for a novel therapy. But I think these new approaches to recognising and treating diastolic function are really urgently needed, particularly in an ageing population and a population where diabetes is becoming increasingly prevalent. So I'm going to touch on the fractal heart. So the inner surfaces of the heart are not smooth. They're covered by this branching network of muscle fibers called trabeculae. So these muscle fibers, they're known to have an important role in the developing heart before the coronary circulation becomes established, because they have a high surface area, and that high surface area helps the heart muscle to absorb nutrients and oxygen from the blood pool before that coronary circulation can take over that job. But they persist into adulthood, and they're really well conserved. They're seen in birds, in reptiles and mammals and so on. The rest of the cardiovascular system is completely smooth, as you would imagine it would need to be to promote efficient flow. But the ventricles, the left and the right ventricles, are highly trabeculated. And really why that is hadn't really been well understood. But these branching, these repeating fractal patterns, so these kind of self-similar patterns at different scales, are seen throughout nature, and it's a very efficient way of specifying development, but also for increasing surface area. On the left here is a sketch by Leonardo da Vinci. He was really interested in these sort of patterns in nature and observed how this branching pattern in trees was optimized the surface area for absorption of light. He was also, of course, a keen anatomist, and he was one of the first to dissect the heart and look at these trabeculae and so on. And you can see here in his sketch these sort of crenellations. This is in the atrium rather than ventricles, but these kind of crenellated surface are trabeculae. So he had a good idea about this. He described them first as being interposed in the furor of the impetus of the blood. So he realized that they were in a critical part of the circulation, and probably did something important. He thought that they created some kind of turbulence in the heart and it warmed up the blood, and that was why humans were warm-blooded, which is an interesting idea. So we did some work trying to figure out what the role of trabeculae were and what the genetic variants that control them are. This is how a certain newspaper interpreted our work, but I think it was nice to kind of link it back to the early anatomists. So how can we quantify this trabeculae? So this is, again, some work from Tim Dawes. It's where we looked at trabeculation in the right ventricle. The right ventricle is very trabeculated. We can create there's the image of the right ventricle. You can see the trabeculae here in grey. We can create like a black and white mask, and then we can trace out the surfaces of the trabeculae here. Now, the more complex the trabeculae, just the greater the amount of surface area on those lines will be. So this is kind of a way of being able to quantify how complex that shape is. So we can basically do box counting. So we change the size of the boxes, we count the number of boxes that touch the surface of the inner surface of the heart, and then we can plot the box size versus the box count, and we get a single number, fractal dimension. This fractal dimension reflects the complexity of this trabeculation. So it gives us a very reliable image-derived number for this complex morphology in the heart. So this is some brilliant work we did with Tim, with Ewan Burney at the EBI, with Hannah Mayers now at Cold Spring Harbour. We did this fractal analysis on 20,000 cardiac MRs in the UK Biobank. We did a genome-wide association study, so looking at these common variants that are associated with how trabeculae develop in the heart. We also looked at knocking out those genes in animal models to see what effect they would have. We complemented that with outcome studies as well, to see if they had an effect on heart failure. And lastly, because it's difficult to manipulate trabecularly experimentally, we used in silico simulations, computer simulations of the heart in a hemodynamic circuit where we could artificially change the trabecular characteristics and see what effect they might have. So a number of interesting findings. It's a heritable trait, trabeculation. It's associated with common variants in cardiomyopathy genes, but interestingly, it's also related to pathways that control actin branching. So actin is a key part of the sort of cell skeleton, and these pathways determine whether a filament is straight or a filament branches. It's also causally associated with heart failure. So we think these trabeculae are an important part of efficient heart function. So in knockout models, so this is where we knock out that particular gene, and this is in a fish model, Medaka, so we can get some idea of what the effect of that gene might be. This is MTSS1, which is one of the genes that we were interested in. We have the wild type at the top, so the normal one, where you can see these branching trabeculae in the heart. In the knockout model, it's very smooth, so the inner surface has lost that characteristic branching pattern. So interestingly, some work published a couple of years before looked at these arborization patterns, these kind of tree-like branching patterns, not in the heart, but in nerve cells, and found if you knocked out that same gene, then you lose that complex pattern of arborization, suggesting perhaps that these genes are a common pathway for the development of fractal traits in different parts of the body, but serving the same purpose to increase surface area for hemodynamic effects in the heart and for information processing in the brain. So this was a great collaboration. Unfortunately, we couldn't celebrate in person, but we didn't let that get in the way. Now, some interesting work from colleagues at the Broad Institute was also based on UK Biobank data, but this time looking at fundal imaging. So this is an image of the retina, the back of the eye. It has blood vessels, which you can segment out, so you can label, and you can also differentiate the arteries and veins. So this is some great work led by Mariam and colleagues at the Broad, and they did a similar to what we did, a genome-wide association, this time looking at these fractal traits in the blood vessels of the eye. Excuse me, they found 7 loci, 7 common variants associated with branching, and these were genes related to angiogenesis and inflammation, but also including a pathway called WNT, which is a very conserved pathway that regulates important aspects of cell fate and cell migration and development of different organs during embryonic development. So I think for us, the really interesting thing was how you can go from clinical imaging to understanding fundamental aspects of cell and organ development. And here, perhaps, getting a glimpse into how very conserved pathways are the same pathways involved in maximizing surface area for a tissue for different purposes. We've now seen it in the brain, we've seen it in the heart, and also in the microvasculature in the eye. So that's the end of the science. I just wanted to mention one of my interests is flying. I've always had a lifelong interest in aviation, and about 10 years ago I decided to challenge myself by learning to fly and got my private pilot's license for helicopters. It was a great challenge, and I think one of the best bits was flying over Sydney Harbour Bridge on an incredibly windy day. But it's been a fantastic opportunity. But it also, I think, just reminds us how it really is good to challenge yourself now and again, to learn new things. And I think actually it's given me a particular interest in aerodynamics, fluid dynamics, which is actually a key part of our research, going to be a key part of our research over the next few years. But actually, it's part of a wider thing. And I think the one thing that I've really learned is summarized by this line from Matthew Syed's book called Rebel Ideas, The Power of Diverse Thinking. And he said that recombinant innovation, so he's kind of coined this term of recombinant innovation, involves fusing together 2 or more ideas from different and previously unrelated fields in order to innovate and develop a true step change in understanding. So I'm a real believer in challenging yourself, in learning new disciplines, in working with some brilliant and really collaborators in very diverse fields. And I guess, not being a cell biologist, being not afraid to ask really simple questions about health and about biology. So with that, I'm going to end. I'm going to thank, this not an exhaustive list, just some of our current collaborators, particularly thank the MRC and UKRI for our funding and the British Heart Foundation in particular, and thank all our brilliant collaborators, our students and clinical fellows over the years for making all this work possible. Thank you very much for listening.
Professor O'Regan, many congratulations. What a fantastic body of work, what a fantastic lecture. Before we move on to the Q&A session, on behalf of your many current, many previous students and collaborators, I'd like to thank you for your mentorship, for your support, generosity, and your endless patience. It's been our privilege to be part of your wonderful journey. We have received many questions today about your lecture, about your work. So I was wondering if we could start with a question from today's live session. We're wondering how important are explainable algorithms when communicating with regulators and the public? And the second question, when is it better to use less complex, more explainable machine learning models rather than more advanced deep learning?
Yeah, I think that's a great question, and in many ways I think the work that we've done has been sort of agnostic to the particular methods that we used. So I guess I touched on at the beginning the sort of conventional hierarchy of different approaches and algorithms. We know that those are going to change; they're not stuck in stone. In 5 years' time, there may be some fantastic new geometric deep learning approaches or other approaches. And it doesn't really change the fundamentals of what we're trying to do, which is trying to improve outcomes for patients on the data that we have. I think explainability is a really interesting thing. I think, we touched on this with our patient group, actually, who are patients with cardiomyopathy or who'd been affected in their families with cardiomyopathy. And how would they feel about these sort of algorithms being used to make decisions about their health and so on? And a couple of key messages, which I think align with the work that we're doing, was actually making sure that people get the right diagnosis at the earliest opportunity for that, because often diagnoses were delayed or missed. Giving people really patient-specific information and advice, particularly around things like lifestyle and exercise, balancing the risk of having a device or not having a device and so on. What they were less interested in particularly was how we get to that point. They wanted advice they could depend on, advice that was specific to them. I think explainable AI is useful to a degree. I think it often comes with the penalty of maybe not having the same performance as algorithms that are less easy to interpret. And I'm not sure that's always a sort of price worth paying. I think it's useful for a sense check and for making sure there are no gross errors and so on. But ultimately, I don't think it's as important as people think. I'm very much a believer in using the least complex, least elaborate approach to solve a problem, and one that's the most robust and reliable. I think a real bottleneck for AI research is just lack of real-world evaluation at the moment. And I think that's really what we're missing. I think explainability, if it can be done without decreasing the performance of an algorithm, that's great. And I think that's useful for clinical adoption and some reassurance about how it's working. But ultimately, I don't think that's really the driver for what patients are interested in.
Yep, thank you. And the next question by Josh Marsh nicely links up. So what is being done, what can be done to safely accelerate this transition from research AI applications to improved healthcare outcomes? And the same, what are the main barriers?
So yeah, I think NHSX, although I think it's wrapped up or been absorbed into NHS improvement now, has really been helpful in this. They've supported a lot of grants at different sort of life cycles of AI development and kind of brought industry and clinicians together to try and, as you say, really make that move between both early stage ideas like our 4D survival algorithm and being able to prototype that for clinical use, but also developing really robust evidence and real world use for more mature algorithms that are perhaps at a closer stage to approval. And of course, the regulatory environment, the approvals process is super complicated. And of course, it's important to navigate that appropriately. So I think it's having the right support, having the right collaborations, and also making sure that you navigate the somewhat complicated territory of medical regulations.
Yep, thank you. Chiara Bicciarelli asks us, how can we use AI in our clinical practice today? And in your view, what's the next best thing?
So I think it's already been sort of transformative, really, in analysing cardiac images. Used to, as you probably will remember too well, analysing these images by hand is, laborious. It's subjective, prone to error, and really AI has genuinely transformed that now. You can pretty much point and click and get results that you rarely need to change. So that has, in terms of time saving, I think Eric Topol's report on AI in the NHS, one of the key messages is how you actually save time for clinicians, how you can avoid them doing things where they don't add value, so they have more time to do things that are of benefit and to be with patients rather than drawing circles on images. So I think that sort of thing has been super valuable. I think AI potentially has value at so many different stages, from image acquisition, accelerating the way in which images are performed, increasing their resolution and contrast and so on, perhaps avoiding contrast medium to make them safer. And we've kind of touched on, I think, the more valuable aspects here of actually stratifying patients more accurately, making sure that the right patients receive the right therapy, not waiting until they've all got severe disease, actually teasing apart those with mild and moderate disease, I think is particularly important, and something that these sorts of approaches, I think, could have more value. So I think, I think there's a whole range of things. I think probably the biggest thing I would guess for cardiac MR in particular is coupled to MR physics, and that's probably getting the acquisition time down to something similar to an echo. So maybe the 10-minute CMR exam, that's a big push from the Society of Cardiac MR. I think AI image acceleration techniques and analysis techniques could be a part of that. But from a patient point of view, from a hospital resource point of view, having a really definitive test that's affordable, that's quick, that's accurate would be a real game changer, I think.
Great, thank you. The next question is more or less tailed off this. How do you think AI will impact the day-to-day work of doctors going forward and their relationship with their patients?
So I would hope, and touching again on Eric Topol's work, I would actually hope that AI, as we've mentioned, actually gives people more time for patients. And I think that even that would be, a fantastic benefit of AI, if nothing else. From our patient feedback as well, I think there's real opportunities for more effective data sharing and engagement, and ownership of data by patients, being able to accept individualized risk profiling as well, particularly for inherited heart disease. I think there are all sorts of opportunities around that. I think there are the more mundane things around workflow and so on. But I think it's really where AI gives you an opportunity to actually spend more time adding value, or the AI itself provides more precision or more rich detail or more value in the patient journey. Those are, I think, the key opportunities.
Yep. And in this world of big data, so lots of our patients will have genetic data available, imaging data, we have their health records. Do you think there's a time in the future we'll be able to predict or calculate the chance of someone getting heart disease, for example, early in their life and advise modification?
Yeah, I mean, I would hope so. And I guess one thing we've sort of seen actually from the pandemic is how perceived risk can change people's behaviour. So even just receiving that kind of change in expected risk, I think, can be a really powerful motivator for behavioural change. even in itself. So you're absolutely right, and I think we touched on one thing, which is of course the very likely increased availability of sequencing tests that are going to be available, of not only people who previously would be considered at low risk, now being discovered that they incidentally have a variant for a significant inherited heart disease, or having genetic screening for some other reason and throwing up these variants. I think that adds a lot of complexity to being able to give them good advice about what it means, and actually be able to reassure them as well that actually having this genetic variant actually may be associated with relatively low risk. Most people with these variants lead normal, healthy lives. So I think part of this is not over-diagnosing people. Part of it is actually giving people reassurance as well, being able to discharge people, not over-diagnosing. So I think there are a lot of really exciting benefits on many levels.
Great, thank you very much. Stuart.
So wonderful. Really, I've seen a number of talks by you, Declan, over the years, and they're always very good, but that was really outstanding. So congratulations for what was really a smorgasbord of wonderfulness. So thank you. I mean, it takes me back, it makes me feel like quite nostalgic. It's like some 15 years ago when we first started working together, and you were the kind of the fatty liver guy and I was the heart guy, and now we've kind of switched roles, and you're the heart guy and I'm the fatty liver guy. So it's funny how research kind of goes that way. But I think one thing that came across to me quite strongly in that presentation was what a polymath you really are. I mean, I think when you look at your trajectory from when you came from looking at tropical medicine, then you did radiology, you did the fatty livers, you moved into the MI cohorts, then created the healthy volunteers, the 3D models, then that progressed into survival, and now integrating rare and common genetic variation. I think you really are bringing across this integration of techniques, which is quite outstanding. So I just say congratulations to you. It's been wonderful to see your journey, and I just wonder where you're going to go next, because you're obviously a leader in the field here, and I think it'll be very exciting to see where you move next. So congratulations, and yeah, really fantastic.
Thank you very much.